Sairam students, welcome to the third session of chapter 11, that is cell structure and microorganisms. So children, before starting today's session, let's revise what we had learnt in the second session. So children, in the second session we had seen two main types of cell, that is the plant cell and animal cell. Right? So this plant cell and the animal cell, they are called as eukaryotic cells. So why they are called as eukaryotic cells? Because they contain nucleus, right? So the nucleus, it has got a double wall membrane. So that's why it is called as a eukaryotic cell. Right? So children, we had seen that in the plant cell there is a outer covering called as the cell wall, which is absent in the animal cell. In the animal cell, the outer covering is called as the cell membrane or the plasma membrane, right? And in the plant cell there is chloroplast. So the chloroplast, they can carry out the process of photosynthesis, which is absent in animal cell. And in plant cell there is a single large vacuole, whereas in animal cell there is a small vacuole, right? So children, after that we saw that there are four main different parts of the cell. That is, first is cell wall, second is cell membrane or your plasma membrane, third is cytoplasm, and fourth is the cell organelles. So just now we discussed that the cell wall, it is the outermost covering of the plant cell, which is absent in the animal cell. Then the plasma membrane or the cell membrane, that is the outermost covering of the animal cell, right? So then we saw about the cytoplasm, the liquid part which is present around the nucleus, that is called as the cytoplasm, right? And the cell organelles, they are actually scattered in the cytoplasm. And the fourth one was cell organelles. So what are these cell organelles? Mitochondria, plastids, nucleus, endoplasmic reticulum, lysosomes, ribosomes, vacuoles, Golgi bodies. So these are the cell organelles. So each cell organelles have got some function. So main part is of the nucleus. So nucleus, it is the important organelle of the cell, right? Because it controls the function of the cell, right? Then the endoplasmic reticulum, right? It is a sprawling net-like organelle, right? So what is the function of endoplasmic reticulum? It makes the necessary changes in the proteins produced by the ribosomes and sends it to the Golgi bodies, right? And what is the function of the Golgi bodies? The function of the Golgi bodies is the proper distribution of proteins, right? And mitochondria, they are also called as the powerhouse of the cell because it produces energy, right? Now vacuoles, what is the function of vacuoles? It throws out the waste products of the cell, right children? So these were the about the cell organelles. Then children, we saw about the microorganism. What are microorganisms? Organisms which cannot be seen by our naked eye and we have to use a compound microscope to see them. Those are called as microorganisms. Now these microorganisms, they are present all around us in the air, water, soil, decaying matter, garbage, everywhere these microorganisms are present. So let's start children with today's session. So here you can see there is a picture wherein it shows variety of microbes. So there are different microbes which are circular in shape, rod shaped, curve form, and there are as well as other shapes of the microbes, right? So now we'll see children that first we'll see about the observation and the measurement of microorganisms. So what you can do children, first is if you want to observe the microorganism, what you can do, you can take a moistened piece of bread, right? Moistened bread means you have to slightly wet it, or you can take a slightly wet bhakri also, right? And you have to keep it in a box, and then after 3 to 4 days you have to open the box, and then you can take a magnifying lens and you can see the fungus grown on it, right? The second example is that you can on a slide you can take a drop of muddy water, and that you can observe it under a compound microscope, or else you can take a drop of the stagnant puddle of water, right? Stagnant puddle of water. So stagnant puddle of water means now in the rainy season the rainwater which is gathered, it does not flow away, right? So after some days you get a dirty and unpleasant smell over there. So that is called as stagnant puddle of water, right? So that also you can take on a glass slide and observe it under the microscope. Then third thing what you can do, you can take a drop of yogurt or buttermilk on the glass slide, and that also you can observe under the compound microscope. So children, generally in the first session only I had told you that these microorganisms, their units are nanometers and micrometers, right? So that's the reason we cannot see them by naked eye. So whatever microorganisms are there, they are smaller in size, that is less than 100 micrometers, right? So that's the reason we have to see them under a compound microscope, right? Then so why do we then what do we do, how to see under the compound microscope? So just now I told you that you have to take it on a glass slide, and then you can observe these microorganisms under the compound microscope, right? So I had told you children that there is a power of the lens, right? So if the magnification of the lens is of 1000x is insufficient, then and you cannot observe the microorganism, then you have to use an electron microscope for seeing it, right? So mostly these microbes, they are smaller than 100 micrometers. So being such a minute microorganism, still there are the organelles which are present in these microorganisms, they can carry out their life processes. Now microorganisms, they are unicellular as well as multicellular. So multicellular microorganisms are which one? The fungus that grows on the bread, or the strands of algae in the ponds, right? So I had just now told you that you can keep the moistened bread in a box, and then after 3 to 4 days you observe it, you will see the fungus growing on it. Then strands of algae means in ponds wherever the water gets collected. Strand means a piece, right? So these algae and fungus, they are multicellular organism. Then there are also unicellular microorganisms. So bacteria and viruses, these are unicellular microorganisms. Now multicellular and unicellular, they have got somewhat a different cellular structure, right? So unicellular organisms, they have got a different cellular structure. So what is different in this cellular structure? They do not have membrane bound organelles found in the eukaryotic cells. So membrane bound means they do not have a outer covering which is found in the eukaryotic cells. So I had told you children, eukaryotic cells means nucleus is present in them. So nucleus, it has got a double wall membrane, right? Then the plasma membrane, cytoplasm and nucleoid are their only components. Such cells are called as prokaryotic cells. So these prokaryotic cells, these are the unicellular microorganisms, as they contain only plasma membrane, cytoplasm and nucleoid. Now children, nucleus and nucleoid both are different, right? Nucleus, as I told you, it has got a double wall membrane, and nucleoid, it does not have any membrane, right? So this was about the unicellular microorganisms. So microorganisms, they are unicellular as well as multicellular. So example of multicellular is the fungus and the algae that grow in the ponds. And unicellular organisms, these are bacteria and viruses. So these unicellular microorganisms, they have got a different cell structure because they do not have membrane bound organelles which are found in the eukaryotic cells. And eukaryotic cells are those which contain a nucleus, as nucleus has got a double wall membrane. So what are the components of unicellular organisms? Plasma membrane, cytoplasm and nucleoid. These are the components of unicellular unicellular microorganisms. So that's why they are called as prokaryotic cells, because they do not have a membrane outside, right? Now this is the figure of the prokaryotic cell. Now you can see it has got a cell wall, it has got a plasma membrane, nucleoid, cytoplasm, ribosomes, plasmid, flagellum and a capsule, right? So this is the figure of the prokaryotic cell. Now how is the growth of the microorganisms? How the microorganisms grow? So the microorganisms, they need specific environment for growth and reproduction, right? They cannot grow anywhere. So some conditions are required. Now some microbes, they need oxygen for their growth, like we require oxygen. But there are some microorganisms which cannot grow, which can grow without oxygen, right? So some require oxygen and some do not require oxygen. Then some microorganisms, they can survive in extreme and adverse conditions. Now extreme and adverse conditions means like the ocean floor. Ocean floor means your bottom of the seabed. Then ice in polar regions, hot water spring. Hot water spring means where naturally the hot water comes, right? So in such conditions also the microorganisms can grow, right? Because we always tell that microorganisms, they need some warmth. So in cold conditions they cannot grow, but there are some microorganisms who can grow in this cold conditions also. But what do they do in these cold conditions? They form a thick covering around themselves and then they stop their life processes for some time. So when the conditions are favorable, that means when that warmth they get, they come out of their protective covering and then they continue their life process. So these microorganisms, what they do in extreme and adverse conditions, they make a thick covering around themselves and they stop their life process for some time. And when the conditions are favorable, that is when they require, when the temperature of warm temperature they get, that time they come out of this protective covering and then again they continue their life process. Now where do microorganisms grow? So they grow in the soil, in water, decaying matter, etc. And what is the temperature for their growth? 25 to 37 degrees centigrade, right? And what nutrition? They require specific nutrients like algae, chlorophyll and oxygen, right? And then what is the atmosphere they require? They require a moist, humid and warm atmosphere, right? Now, according to the shape and the life processes, the microorganisms, they are classified as algae, fungi, protozoa, bacteria and viruses, right? So I had told you there are different shapes of the microorganism, right? And depending upon their shapes, their function is also decided, right? So depending upon the function, their shape is there. So and for carrying out the life processes also, what they are classified into these categories: algae, fungi, protozoa, bacteria and viruses. Now children, in Pune, there is a national institution called as National Center for Cell Science, that is NCCS, right? National Center for Cell Science. So this institute, it does research in the cell processes, that is called as cytology or biotechnology also, right? Now what are the useful microorganisms? Now children, there are harmful microorganisms also and useful microorganisms also. Now these useful microorganisms, they are present in the soil, dung, etc., right? So how they are useful? They decompose the garbage to obtain the food material, right? And this garbage, it is converted into manure of the best quality and the surroundings are kept clean. Also for proper sewage disposal, sewage means I told you the bathroom water or the toilet water, there also what is done, microbes are released into the sewage so that it helps in the quick decomposition of the organic compounds in it, right? So I can give you an example children. like you can take two earthen pots mix in which soil is added, right, and you name it as A and B. So in the A earthen pot, what you can do, children, you can add this fruit peel, vegetable stalks, paper scrap, dung, etc. And in the second pot B, where you have added the soil, in that you are going to add metal scrap, then pieces of glasses, etc., right. And then after some days you can observe it. So when you observe, you will see that in the earthen pot A, wherein you had added the fruit peel, the vegetable stalks, all these, so that gets converted into a manure, right. So this manure, it is of the best quality, right, and that you can use it for your plants. So as I told you, useful microorganisms, they are present in the soil and dung also. So what do? They decompose the garbage and to obtain the food material, and this garbage, it is converted into manure of the best quality, and our surroundings are kept clean because of it. So also for sewage disposal, proper sewage disposal, also microbes, they are released into the sewage, so that quick decomposition can take place. Now some microorganisms, they are present in the soil, and those in the root nodules of the leguminous plants, that is pea, bean, etc., and they convert the atmospheric nitrogen into its compounds. So children, in the chapter 4, that is nutrition and in living organisms, we had seen that the in the atmosphere gaseous nitrogen is there, which the plants, they cannot take it directly. So what happens, there is a bacteria called as rhizobium, which is present in the leguminous plants, and it helps to convert this atmospheric nitrogen into the compounds. Also in the soil, azotobacter microorganism is present, and that also helps to convert the atmospheric nitrogen into the compounds. Now these nitrogenous compounds, what it does, it helps to increase the fertility of the soil, and also the protein content of the pulses also increase, those which are present in the soil. So microorganisms, they are helpful in converting the atmospheric nitrogen into its compound, and because of this, what happens, the soil fertility is increased, and the protein content of the pulses also increase. So children, this was what in today's session. So let's recap what we have learnt in today's session. So children, in today's session, we saw about the microorganisms, right? Like microorganisms, they are unicellular and multicellular. So we saw the example of multicellular, that is the fungus and algae, and unicellular, that is bacteria and virus. Now these unicellular microorganisms, they are prokaryotic cells, because they do not contain nucleus. They contain plasma membrane and your nucleoid and cytoplasm. They do not have a double wall membrane around them, so that's why they are called as prokaryotic cells. Then I told you children that there is a institute called as National Center for Cell Science in Pune, which does research in this cell structures, right, and that is called as the cytology and biotechnology also, right. Then children, we saw about the growth of the microorganisms, how where do these microorganisms grow, right. So these microorganisms can grow in adverse and extreme conditions also. So during extreme and adverse conditions, what they do, they make a thick outer covering around themselves, and then they stop their life process, and when the conditions are favorable, they come out of this outer covering and they carry out their normal life process. So they require a temperature of around 25 to 37 degrees centigrade, right, and some microbes, they require oxygen for their growth, some do not require oxygen, right. Then we saw about the useful microorganisms. So there are useful microorganisms also and harmful also, right. So useful microorganisms, what do? They convert the garbage into manure, and and then we can use it as a best quality manure for our plants, right? Then some microorganisms, as I told you, they like your rhizobium, which is present in the leguminous plants. They convert the atmospheric nitrogen into the compounds, and then the plants, they use it for their growth. Also in the soil, azotobacter microorganism is present, so it also does the same thing. It converts the atmospheric nitrogen into the compounds, and what happens because of this, the fertility of the soil increases and the protein content in the pulses also increases, right children? So in the next session children, we'll see about the harmful microorganisms and some more thing, right? Till then, goodbye.